Anyway, so antibiotic resistance. Now, as the majority of you are going to be future, healthcare specialists, this is something that you can deal with a lot. And before we talk about the mechanisms of resistance, we need to figure out how it can arise in general. So first, I want you to understand that not all microbial resistance to antibiotics comes from human actions. There is so-called intrinsic resistance. The one that we discussed before, Penicillium notatum, is killing its competitors. So its competitors, during the process of selection, under that selective pressure, acquire a phenotype that is resistant to whatever Penicillium notatum produces. Does that make sense? So, depending on the environment that microorganism is normally existing in, microorganism may have normal, natural, intrinsic resistance to penicillins or cephalosporins or aminoglycosides. All those antibiotics are natural or semi-synthetic. Does that make sense to you? Now, on top of that, so that is selective pressure. So first of all, we need to say this. So selective pressure leads to the development of antibiotic resistance. And this is really an important statement because this is the phenotype that doesn't sporadically appear. You have to understand that adaptation like this, resistance to something, comes at the cost, energy cost, nutrient cost, some kind of a cost. Does that make sense to you? Now, how do we humans contribute to the antibiotic resistance? When you see or hear the statement, use of antibiotics, makes bacteria resistant, no, it doesn't make them resistant. It selects for the ones that are already resistant. In any population of microorganisms, you're going to have bacteria that have different sensitivity to antibiotics. Resistance can be represented as the scale. Less resistant, more resistant. Does that make sense? Some of them produce high levels of beta-lactamases, others produce none. We good? Is that understood? And another important thing that you have to appreciate, when we say microorganism is resistant, doesn't mean it's like Incredible Hulk that cannot be destroyed by anything, right? It means that normal conventional doses of antibiotics are not effective against it. You can still blast it with, 10 times higher dose and kill it, but here comes the question, what about a patient? If you do it in the cell culture or in a plate, it's fine, but if you try to increase the antibiotic dose in a patient, that's a totally different story. Makes sense, right? So that's the scale. So what drives the resistance? Two things. First, medical use of antibiotics. And we shouldn't blame doctors. We should blame patients. Two main sources from patient side. First, lack of compliance. Okay. And I must say, I'm an example. Nobody is perfect. We in the family were an example, and we were like a typical example of poor compliance. My son got strep throat. He got antibiotics. And I thought that he's 13, so he can take the whole course by himself. I was wrong. He missed 2 last bills. 2. Out of 7-day course, I think so. Last day he didn't take. Guess what? 2 weeks later, he has a recurrent strep throat infection. And that time, he was on antibiotics for like 2 weeks or something like that. Okay? So that's lack of compliance. To my exoneration, I can say that it was pretty mild lack of compliance. Sometimes people stop taking them the moment they feel better, which is totally wrong. The idea in taking antibiotics is that you launch the assault on bacteria and you clear the battlefield completely. You nuke them, you wipe them out, even the most resistant ones. Because which ones will die first? Less resistant, right? And then if you stop taking antibiotics after that, more resistant will stay, persist, and can cause recurrent infection. That's number 1. Number 2, from the patient side, there's a perception that if you go to the doctor with some kind of a disease, you got to get a pill. Sorry if it insults anyone, but people are stupid. As one Texas lawmaker recently clearly demonstrated, saying that measles is not a problem in the United States. We have antibiotics. Bullshit. Measles is the viral infection. Antibiotics are not efficient against measles itself. They do help to treat complications, possible complications of this totally vaccine-preventable disease. But measles is not treated by antibiotics. It's not treated at all. It can kill you. So when people come to the doctor and the doctor says, sorry, you have a virus, there's nothing I can do. You have common cold. Go home, drink fluid, don't go to work, and get better. People sometimes are displeased because they don't get the pill. Which, some doctors, they will buckle under the pressure and say, just take this and go. There was suggestion, just prescribe the fake pills, like sugar. Huh?
Vitamin C.
Say again?
Vitamin C.
Sure. Yeah, vitamin C. Of course, no problem. Okay, not gonna, well, if you don't have any adverse reactions to actual this powder or whatever they sell, sure. Okay. Does that make sense? But probably the more, and in light of this, now there's antibiotic stewardship program that was initiated by various societies in the world. For instance, American Society of Microbiology actively promotes it. It actually encourages doctors to prescribe antibiotics more carefully, which is great on the paper. Sometimes you should be your own advocate. Not, insisting on give me the pill. But, be reasonable. If you come to the doctor and you have some obviously pyogenic respiratory infection with a greenish sputum, and you're being told that's family's experience, and you're being told, oh, you have an allergy? No, I don't have an allergy. I know what allergy looks like. It's not an allergy. Do the tests, for that matter, to, figure out if it's the allergy or bacterial infection. Does that make sense? So you have to be your own advocate. In the end, whoever it was recovered from that quote-unquote allergy, but it was pretty bad experience. We had other experiences with not prescribing antibiotics later, which I don't want to share. It was pretty bad. So you have to advocate. You have to ask, can you please do the test? Can you rule out bacterial infection? Okay? You have to be your own advocate. The big source of antibiotic resistance is agriculture. Okay? in agricultural settings, we used to treat cattle and poultry with antibiotics to improve growth, especially poultry. And after outbreaks in Netherlands, there was a huge outbreak in Netherlands that actually killed several people. It was identified that antibiotic resistant E. coli came from chicken. Europe, they took measures to eliminate antibiotics from agriculture completely, and they did. Use of antibiotics is banned in Europe, and as far as I know, vast majority of agricultural manufacturers in the United States, they abandoned antibiotics because it doesn't improve growth that much. So it's kind of logical. Okay. Now, how microbes can actually evade the action of antibiotics. This is what I like about microbiology as microbiologist, and don't like about microbiology as an instructor. 2 months ago, the paper was published showing 8th mechanism of escaping the actions of antibiotics. So now, for the next semester, I need to update the table again. First mechanism, I'm going to tell you about the 8th. It's not going to be on the exam. I'm just going to describe what it is. First is drug modification. Okay? That's beta-lactamase. Beta-lactamase modifies a drug, modifies beta-lactam. Does that make sense? Penicillinase opens up beta-lactam ring in penicillin. Cephalosporinase opens up the beta-lactam ring in cephalosporin. So it chemically alters the drug. A pretty obvious mechanism, prevention of cellular uptake. Drug cannot get in. Right? Efflux pumps. The microorganism pumps the drug out of the cell. Is that clear? Now, this is a great example of resistance that comes with a cost. Pump. Does it require energy? Yeah. So to pump the drug, microorganism has to use or produce more ATP, right? So it does come with a cost. Target modification. Now, that's the great story. So, say you have a target that is penicillin binding protein. So penicillin can bind to it. Now imagine that that penicillin binding protein slightly changes. It still can form peptide links, but penicillin doesn't bind to it anymore. So target is modified. In fact, in methicillin-resistant Staph aureus, the main mechanism is target modification. The PBP, the transpeptidase in MRSA, is changed so methicillin cannot bind to it and cannot inhibit it. Does that make sense? Target overproduction. Now remember we talked about sulfa drugs, how they inhibit individual steps of the metabolic pathway. Does that make sense? Now imagine you have, hypothetically, 5 molecules of sulfonamide and 5 molecules of the first enzyme, the hydroxypyruvates synthase. If you have 5 to 5, the likelihood is all 5 are going to be inhibited, right? But if you still have 5 molecules of sulfonamide and 5,000 molecules of that enzyme, you inhibit 5 of them, but 4,995 enzymes are still fully functional. Does that make sense? So in response to antibiotic, maybe microorganism can increase the production of the enzyme, transcription, translation, synthesis of enzyme. So drug will not inhibit all of it. Again, it comes with a cost. You have to produce more enzyme. Does it make sense, this idea? enzymatic bypass. A different pathway is developing or developed that allows the microorganism to achieve the same biosynthetic goal. Kind of a detour, enzymatic detour. And target mimicry is one of my favorites. So, cephalosporin, one of the fluoroquinolones, binds to DNA gyrase. Remember that, right? Some microorganisms produce peptides that look just like DNA gyrase. So, antibiotic binds those peptides instead, and gyrase stays unaffected, and everything works. It's kind of a decoy, okay? False target mimicry. Yes.
But then, so it wasn't made in that way. It was made as just like some kind of defective protein, and that just happened to be a mutation that
probably so
worked.
Probably so. Think about this: that mutations are random always. They never directed, but the process of evolution is never random. So we have, if under that selective pressure, having this short peptide turns out to be advantageous, then those microorganisms are going to survive. You have to understand, you remove that selective pressure, it may be a disadvantage because you produce that crap that nobody needs. It has no functional meaning, So you have to use some energy on it, right? So yeah, it's absolutely possible. Now, the eighth mechanism that I actually learned just recently about was found in E. coli. Just for your information, it is called mini cells. Now, the existence of mini cells has been known for a long time. So what they are, if you would look under the microscope at the population of E. coli that are exposed to some kind of environmental stress, including antibiotics, you will find that in that population you have functional cells that have DNA. Are you with me so far? And you also have what looks like tiny little pouches, like cells, mini cells, that have no DNA whatsoever. And the role of mini cells wasn't clear at all until recently. Turns out that E. coli, that specific phenotype of E. coli that can form mini cells, they use mini cells as dumpsters. They package all antibiotics into the mini cell. The mini cell detaches from the main cell. And now you have, it's like a trash bag with all the crap that cell doesn't need. It was very convincingly shown that cells that can make mini cells, E. coli that can produce mini cells, they have much higher level of antibiotic resistance than the ones that can't. Okay, I think aminoglycosides were tested. So they can basically package those aminoglycosides in these trash cans, just throw them away. Okay? And a great example, actually, that same group, they compared the growth of a regular E. coli and mini cell producing E. coli without antibiotics. Guess what? Regular ones grew better because they didn't have to spend and waste energy on making those mini cells. Does that make sense to you? So, resistance comes with a cost. Now, in regards to this, what you need to know. Match the term and the definition. I give you a description of a mechanism. You identify the mechanism. Okay? On the scale of how hard the question is, from easy to hard, I put it on medium. It's not super easy, but it's nothing like Ames test question that was on the second exam. So it's medium. here, what you need to know. I give you this 6 different acronyms for most common antibiotic-resistant microorganisms. You need to know what they stand for. For instance, VRE is vancomycin enterococcus, okay? Or CRE is what type of bacteria CRE is? Enterobacteriaceae. Does that make sense? So you need to know what they stand for. It's all here. You do not need to know the numbers. But you just, marvel at those numbers, really. No, seriously, just look at them. Here. Vancomycin-resistant Staph aureus now kills 11,000 people a year. Okay? Vancomycin-resistant enterococcus is responsible for 20,000 hospital-acquired infections. So we're talking about 150,000 cases, about 150,000, only for the ones that I present here. Think about how much it costs to treat a person. Hospital stay, drugs, all the procedures. So we potentially talking about billions of dollars in medical costs to treat antibiotic-resistant infections. Now, few words about each of those. So first, MRSA. Methicillin-resistant Staph aureus. We talked about it so much, it doesn't need any additional presentation. Hospital-acquired infection. Resistant to beta-lactams, methicillin. When MRSA appeared, the second drug of choice for treating gram-positive infections and staphylococcal infection became vancomycin. Well, guess what? Now we have vancomycin-resistant Staph aureus. And on top of that, wide use of vancomycin in clinical practice led to development of resistance in normal human, commensal human microorganisms, enterococci. So now we have vancomycin-resistant enterococci that regularly cause wound infections, lung infections, urinary tract infections in hospital patients. Now this tongue-breaking acronym, ESBL, stands for Extended Spectrum Beta-lactamase. So this gram-negative bacteria, let's admit, it's mostly colonic microorganisms like E. coli, Enterobacter, Klebsiella, Proteus, not Clostridia, no. Different Bacteroidetes, like Bacteroidetes fragilis. So these gram-negative microorganisms have beta-lactamases that can neutralize various types of beta-lactams, from penicillins to cephalosporins to monobactams. Thankfully, this extended spectrum beta-lactamases do not neutralize carbapenems, our last hope. Unfortunately, several years ago, first reports came out of India about a discovery of carbapenem-resistant Enterobacteriaceae, specifically carbapenem-resistant Klebsiella. And that was really a bad news. Because I mentioned carbapenem is our last resort, nuclear option antibiotic. Okay? And it turns out this gene of carbapenem resistance was found in the plasmid, which is another bad news. Easy transfer. Okay? And for instance, there was a case, a woman came from India. She developed a systemic infection, Klebsiella infection of kidneys, and she had to spend several months in the hospital for inpatient antibiotic treatment of kidney infection with carbapenem-resistant Klebsiella. Again, resistance doesn't mean that it cannot be killed, okay? But it's super hard. So they had to do the combination therapy. It was a long therapy. They had to keep her in the hospital in case of complications and side effects and so on and so forth. She was released. She recovered completely and was cleared. But look, 4 months for kidney infection, it's a long time. Usually UTIs, we're talking about a week or two. 3rd generation cephalosporins, and you're out. Okay? Now, it is a concern, of course, but the main concern actually, well, much more important, I would say, is Mycobacterium tuberculosis. So these acronyms MDR, XDR, TDR stand for multiple drug resistant, extensively drug resistant, or totally drug resistant. So totally drug resistant mycobacteria can be resistant to everything, from kanamycin, which is pretty old, to isoniazid, which is fairly new. Okay. This is why you notice when we talked about antibiotics, I mentioned dialquinolones can't treat mycobacteria. Rifamycin can't treat mycobacteria. Isoniazid can't treat mycobacteria. It's a problem. Okay. The number of cases may not be huge, but tuberculosis, as we will see, as we will discuss on Tuesday, is extremely contagious. Okay? And there was a case when an Indian gentleman was diagnosed with either XDR or TDR, Mycobacterium tuberculosis, extensive or totally drug-resistant one, and he was strongly advised not to go anywhere. Basically, take the drugs. He had prescriptions for a bunch of antibiotics. Take the drugs, stay home. He went to India instead for wedding of his brother. And then he returned back, was apprehended, not like criminally apprehended. He was brought to the hospital and was treated in the hospital. And every person on every flight had to be checked. Thankfully, nobody got infected, but still, what if somebody would? It's not just common cold or even measles. It's tuberculosis, which is incredibly hard to treat. Okay? As far as I know, the gentleman got treated. And India, again, I mentioned India. The problem is that the poorer folks in India often cannot afford a full course treatment for tuberculosis. And India, one of the leading countries in terms of the total number. Percentage-wise, it's South Africa, but total number, India, China, huge populations are positive. So if people cannot afford, say they go to the regular physician says, sure, here's your 4-month, therapy with this, then that. The person looks at the bill and says, holy crap, I can't do this. Okay? And then this person goes to sort of a local health practitioner, not physician, I don't know how, not as sophisticated. And the person says, sure, here's your prescription for kanamycin, 2 weeks, you're going to be fine. So the folk goes to the pharmacy, gets the drugs, takes for 2 weeks. Getting better? Absolutely. Feels much better. On top of that, he's now got drug-resistant tuberculosis. And that happens pretty frequently just because of the sheer population size. Okay? And a lot of new drug-resistant infections, tuberculosis especially, they actually happen to arise there. Make sense? Okay. So, we're going to stop at this point.